My name is Christian Nelson. I lead sales and support here at Arima-Genomics, and I'll be your host and MC for today's webinar. Agenda for the day, we'll start it off with our Senior VP of Science, Anthony Schmidt, who will be talking about the background, base platform of Arima-Genomics technology and Hi-C. And then we'll move on to our keynote speaker and then questions at the end. Let's get to it. So our first speaker of the day is Anthony Schmidt, who has served as our Senior VP of Science since 2016. He has led the development of the core Arima-HiC chemistry and technology platform since Arima's inception. He's contributed to the core inventions of technology development grants and continues to lead our R&D programs for targeted chromosome conformation capture techniques such as HiChip and Capture Hi-C. He's got a lot up his sleeve for future product launches, so please keep an eye on your inbox later this year. He's well-trained, well-published in the field, and a generally good person to work with. Anthony, the floor is yours.
All right, excellent. Thanks a lot, Christian, for the introduction. Just wanted to turn on the video and say hi to the audience and thank everyone for attending before I get started, and I'm gonna shut off my video. So welcome. So today, I'll just be giving a quick primer really on sort of the basics of Arima-HiC technology platform, and then just share a couple of quick vignettes on applications in translational oncology. Obviously, we're going to hear a lot more about that from Giorgio, but I just wanted to sort of seed a couple little stories on how we're excited about using our tools when it comes to oncology research. So with that, just a little bit of background about Arima. Arima is a biotech company based here in San Diego, California, that aims to accelerate the understanding of what I'm going to call 3D or three-dimensional DNA folding within cells. And in doing so, transform discovery, diagnosis, and therapy. And we really try to facilitate access to this high-quality 3D DNA folding information via kits and services. And just a little bit of background, this is what the Arima-HiC kit looks like. We launched this back in 2018, really geared towards genome-wide 3D DNA folding analysis. And of course, over the years, we've received lots of feedback about how genome-wide tools are not always the most appropriate to address particular biological hypotheses or applications. So since we've launched two different forms of targeted DNA folding analyses, namely HiChip and Capture Hi-C, and I'm going to talk about what delineates these three types of assays in just a minute. So with that, just a little bit of an outline here. First, I'm going to talk about the technology platform and the three different approaches, and then tell two quick vignettes on applications in translational oncology through the lens of customer or Arima co-authored publications. But first, a little bit of background on 3D DNA folding. This type of approach has been used to study gene regulation or gene misregulation across a whole host of different biological processes, and I don't have time to go into all of them, but here's just a representative set of publications that have used Arima tools across stem cell biology, immunology, cardiology, as well as applying these to developmental disease, neuropsychiatric disease, as well as cancer, which we'll dive into more in the second half here. So a bit about the technology platform itself. I like to start at the highest level possible. I mean, we think of these tools as a means to read the DNA folding within cells. And at the most sort of basic workflow level, the first step of that process is what we call Arima sample prep. And during that, you essentially capture, using molecular biology, the three-dimensional folding within cells. And what comes out of that is just extracted DNA, right? So it's sort of like a glorified DNA extraction approach where you preserve 3D DNA folding information during that extraction. That then flows into an Arima library prep, followed by sequencing. And then on the back end, Arima bioinformatics, so that you can make sense of that 3D DNA sequencing data that you've obtained and apply it to your particular use case. And to peel back a layer of an onion, just one layer here, and show sort of the nuts and bolts of the technology for those who are interested. At the very beginning, what are the compatible sample types? Well, here's just a smattering of different sample types which are possible for the Arima-HiC platform. Of course, blood, cell lines, frozen tissues, FFPE tissues is a capability that we've been exploring, and I'm actually going to tell a story about Hi-C technology towards oncology research that used FFP tissues in the second half of this presentation, as well as insects and plants. Okay. So fairly flexible input requirements here. And then this is really the core Hi-C chemistry, starting with fixed cells. So if we're looking down at an interphase nucleus here, you see two pieces of DNA that are gray and blue that are spatially proximal. So they're held together close in three-dimensional space. And if I just sort of zoom into one of those blue and gray pieces that are held together, this is what we're looking at, right? So we have protein holding these two pieces of DNA in close spatial proximity. And then there's really just a few core steps to capture that three-dimensional proximity. The first step is to cut the genome up. We use a cocktail of restriction enzymes. So we cut the genome up, then we label the ends with a biotinylated nucleotide, which you'll see why we do that in just a moment. And then we ligate the ends back together, and then we purify the DNA. So these proteins go away. So the two pieces of DNA, blue and gray here, that were spatially proximal are now physically joined together, and they're linked at this ligation junction that's marked by a biotinylated nucleotide. We have a predictive sort of quality control assay here that comes right here that I don't have time to go into now, but we have that workflow QC as well. On the back half of the workflow, we shear the DNA into smaller pieces so it's compatible with Illumina sequencing. And then we enrich those informative molecules, the ones that are part gray and part blue, using biotin streptavidin conjugation. So that carries through only the ones that contain information about blue and gray, which are the chimeric molecules, into library prep, PCR, and then paired end sequencing. So when you sequence this one particular molecule and you align it back to, let's say, a human reference genome, the gray piece will map to where that originated in the genome, and the blue piece will map to where that originated in the genome. And that is effectively one data point from one cell that those two pieces of DNA were in close spatial proximity. And like I was saying in the beginning, we've since developed and launched ways to capture this information in a targeted fashion. So similar to how you would do exome sequencing, for example, you can apply target enrichment to Hi-C libraries. Sort of an analogous approach where you just design complementary oligos that would hybridize, and you can enrich any real sequence element of interest. So think promoters, all promoters in the genome, or just a subset of promoters that relate to genes that are interesting for your particular research, GWAS variant loci, or other regions of interest. We have lots of content from past webinars, as well as application notes about these different forms of targeted Hi-C. You could also enrich at this stage here, when you have proximally ligated chromatin, you can do a protein enrichment using an antibody. And why you might want to do that would be if you want to basically elucidate those three-dimensional DNA folding interactions that were specifically associated with a protein of interest. So think K27 acetyl, CTCF, cohesin, or other histone PTMs or transcription factors. All right, so we have genome-wide and targeted options. So that's really the core, molecular biology. And like I said, we have bioinformatics tools at the back end and a pretty hands-on tech support model to help you be successful with the assay and the informatics. Okay, so the first application I want to talk about in oncology comes from this customer publication. This is from the Iofantis and the Syragos labs at NYU. What we're looking at here is a Hi-C heat map from healthy T cells. So the dark red color means that there's a high spatial proximity between any two regions along the linear genome, which is along the diagonal here. So the MYC gene is located right here in this highly interactive domain, right? And then about 2 megabases or so downstream are a series of super enhancers that are in their own. You can sort of see this faint red domain here, but very little interaction with the MYC gene. So for those who are not used to looking at Hi-C heat maps every day like we are, that could be represented kind of like, here is the MYC gene over here, here are here's the enhancer element over here. You can tell that they're spatially segregated into separate but neighboring topological domains. When you do these types of Hi-C analyses in T-cell acute lymphoblastic leukemia samples, the topology around the MYC locus is drastically different. So here's a primary T-ALL patient, and here's a leukemia cell line. You can see that it's vastly different. This well-defined domain that was present in the healthy T cells has sort of dissolved into the next domain. So they've sort of fused together into one larger DNA loop, which is shown here. And with that different topology comes an increase in MYC expression by this little green line, as well as transcription factor binding at the super enhancer elements. And of course, here's just a quantification of the MYC expression in the leukemia cells versus the healthy primary T cells. I think this is a great example because it really, I think, illustrates the value of looking at the 3D genome and finding these types of 3D genome folding biomarkers and how they help you interpret disease mechanisms. So if you were doing this type of study just with, as we would call it, linear assays without this three-dimensional context, you would see in a healthy individual, here's the MYC gene, 2 million base pairs away are some super enhancer elements that are not active. And then in the disease state, the MYC gene turns on, you see transcription factor binding 2 million base pairs away. And then when you treat, maybe this turns off and you see a reduction in transcription factor binding. But in the 3D genome paradigm, it really informs how MYC is regulated in the healthy state, as well as how MYC is regulated, or I should say misregulated, in the disease state. I think what's important about this study too is through the joint analysis of Hi-C as well as ChIP-seq, and knowing that this particular transcription factor binds to this enhancer element and it's spatially proximal to MYC, gives the researchers some clue that targeting this particular transcription factor, which in this case is CDK7, may have a therapeutic benefit in reducing MYC expression. And that's exactly what they found out through their work. So I think this is a great example of how 3D DNA folding can sort of define elements of the disease state and maybe even inform how. This is the same image that I just showed, but I actually wanted to throw up a poll question that I showed before. The question here is, if you were only interested in profiling that three-dimensional folding information in just the 3 megabases around MYC, and nothing else in the genome, you didn't care about protein-mediated interactions, but just the 3 megabases around MYC, which approach would you take? For those who have attended our previous oncology webinar, I did show this question before. So this is maybe a test to see if you remembered, and then for those who are new, hopefully this makes sense. We'll give maybe another few seconds, and then we can show the poll results. All right, let's show those results. Capture Hi-C, excellent. Yes, that is at least what I was thinking would be an appropriate approach. And what I've shown here is just tiling those captured probes across the MYC locus and being able to enrich for those interactions across the MYC locus. Excellent. So in the last couple of minutes here, I'm going to talk about a different application in oncology, specifically identifying novel drug targets. Through the lens of this collaborative manuscript we had that was led by Lucas Chavez at UCSD, that we got brought into really because of our FFPE Hi-C capabilities and how there was only tumors in FFPE format, but really needing that three-dimensional folding data to help put this story together. So I'm just going to dive right into it. This is just a tiny part of this particular paper, but there's a particular subtype of ependymoma called REL-A ependymoma, and it's characterized by a gene fusion between REL-A and C11 or ORF95. So if you look at just the three-dimensional topology in the 1 megabases or so around the breakpoint. Here's the relay gene. You can see it's right here at the break. This is what it looks like. If you look at C11 ORF95, this is what the topology in the megabase around it looks like. These two are fused together, relay C11 ORF95 fusion. And what's really interesting about all fusions or all translocations is that the rules of 3D genome topology apply to the fused chromosome. So you get these high-C interaction patterns or these DNA folding patterns that happen across the breakpoint, which sort of enables potentially inappropriate interactions between enhancers and genes across the breakpoint that wouldn't normally be there in the native unrearranged context. So what we're really talking about here is an event kind of like this, where on the relay side, you may have a enhancer. Here's the fusion gene interacting across the breakpoint with a gene here called RCOR2, and that gene is then expressed on the rearranged allele. So that's ultimately what we observed, and we asked the question here: is RCOR2, this gene that undergoes enhancer hijacking, essential for relay attendance? So that gene, or at least the transcripts, are essential for relay ependymoma cell survival. The next question then being, well, we know that RCOR2 forms a complex with a protein called LSD1 as well as HDAC, and can that pathway or protein complex be targeted to have some sort of therapeutic benefit in this specific subtype of ependymoma? So since there's already HDAC inhibitors available, you apply an HDAC inhibitor to a patient-derived cell line and in fact observe tumor cell death after application of the HDAC inhibitor. So this example that I told you is really in pediatric ependymoma, but we've had other customers publish different stories in different cancer types, including bladder cancer, prostate cancer, pediatric high-grade glioma, as well as pediatric leukemia and others. So in conclusion, we believe that 3D DNA folding is a really important dimension in translational oncology research by the two applications that I just told you, as well as others that you're going to hear in just a moment from Giorgio. Hi-C is a powerful technique. It's a sequencing-based approach to measure that important 3D DNA folding information. And lastly, we believe that our technology platform is a really nice, robust, but flexible toolkit, whether you want to go genome-wide for discovery or some form of targeted analysis. So that's kind of how we think about our tools. And with that, I'll conclude and thank you all for your attention and pass it back to Christian, I think, to introduce Giorgio.
Great. Thanks, Anthony. Always appreciate the overview to the Arima-Genomics Hi-C platform. Moving on, we have Dr. Giorgio Galli, who's joining us from Novartis out in Basel. We appreciate your time, especially being a little late in the evening out there, Giorgio. Just want to give you a quick little introduction, and I'll pass it over to you. Giorgio obtained his PhD at the Biotech Research and Innovation Center and University of Copenhagen in Denmark, working on the molecular functions of the transcription factor PRDM5 and embryonic development in cancer. He then did his postdoctoral training at Children's Hospital in Boston and Harvard Stem Cells and Regenerative Biology, working on hippo pathway transcriptional complexes in adult stem cells in cancer. In 2015, he set up his lab at the Novartis Institute of Biomedical Research in Basel, Switzerland, in the oncology department, focusing on large-scale functional genomic screens. He then became group leader in the oncology drug discovery biology unit. His laboratory focuses on characterizing the molecular mechanisms engaged by transcription factors and chromatin remodeling complexes to drive novel drug discovery programs. Giorgio, thank you for your time. Welcome.
Hi, everyone. Good morning, good afternoon, good evening, according to where you are. Thank you, Christian, so much for the kind introduction, and thank you, Arima, for the invitation. So what I want to do today is to give you the highlights of a story that we recently published on our efforts to try to characterize how transcription factors engage non-coding regulatory elements in the cancer genome in order to understand how these proteins exert their proliferative oncogenic program. So, sorry, just a second. Okay. So over the years, we heavily invested in performing functional genomic screens in order to identify cancer dependencies. Some of you might recall the DRIVE project, in which we screened 400 cell lines by a genome scale shRNA library, where we identified several known targets, mostly genetic drivers, as well as new oncogenic, new oncogenes involved in either metabolism, synthetic lethal relationship. And one of the prominent category of hits we identified from this kind of screens were what we call expression drivers, which are essentially lineage-specific transcription factors, which are basically transcription factors important. You can see, for example, here, SOX10 and MITF specifically expressed in skin, particularly melanocytes, and cancer cell lines derived from this lineage, they are sensitive to the knockdown of these factors. And the same goes on for several other transcription factors in different lineages, for example, MIB in heme malignancies. So we became very interested in trying to understand how these proteins are working in cells and really to go deep into their proliferative oncogenic program. So one of the transcription factors we focused on is really a prototypical oncogenic transcription factor, which is estrogen receptor. We do know that in normal breast, and particularly in estrogen receptor positive breast cancer, the gene ESR1 is expressed, and when we knock down ER, which is the protein name of ESR1 and its cofactors FOXA1, GATA3, we know that cancer cell lines are sensitive to the knockdown of these proteins. And indeed, this was well established, and we know pretty well how to target this pathway. And this is because estrogen receptor is a transcription factor that senses extracellular stimuli, such as its ligand, estrogens, or RTK signaling in order to get activated. Once it gets activated, it dimerizes and enters into the nucleus. And either it binds directly DNA, recognizing its own binding motif together with its co-activators, or by engaging other transcription factors, it binds to a number of regulatory elements and drive transcription of proliferative genes. Additionally, one of the genes that we're particularly interested in downstream of estrogen receptor is cyclin D1, because this is a cyclin dependent, it associates with cyclin dependent kinases, which can be targeted by CDK4/6 inhibitors. So we do know pretty well how this pathway works. We do know how to inhibit the functions of estrogen receptor by preventing its activation. And we know how to target downstream cyclin D1, one of its critical target gene. What we didn't know, and we decided to invest in, was to understand how can estrogen receptor and cyclin D1 stay expressed in this cancer? So what are the regulatory elements that are sustaining their expression? And how do they contribute to the oncogenic phenotype? So to address this question, we decided to use a CRISPR-Cas9 approach. So we first generated reporters for estrogen receptor and cyclin D1 by knocking into their endogenous locus a GFP tag so that we could monitor their expression. And then using Hi-C data, we defined the topologically associated domains that delimit the loci of ER and cyclin D1. And we brought to essentially every possible DNA hypersensitive site, so any accessible site ever found in 95 cell lines, a dead Cas9 fused to a repressive element, and this system is called CRISPR-I, in order to repress one by one every regulatory element ever discovered in the loci of ER and cyclin D1. And so we transduced MCF7 breast cancer cell line with this custom library targeting every regulatory element in the locus. And we could monitor both ER or cyclin D1 expression by using the GFP reporter, or looking at an enrichment score, because when we affect these genes, both ER and cyclin D1, we know that cancer cell lines will die. And so this is just how to give you a flavor on how this data can look. This is the locus of estrogen receptor in red here. This is a genome browser snapshot where you can see the ER gene in red. And on this track here, which we call ODBC proliferation, you can see a moving window of the scoring of all these gRNAs in the locus in terms of how much they affect proliferation. Additionally, we can monitor, as I said, the GFP expression, which reflects estrogen receptor expression. And you can see that in this whole locus, the guides, there's an enrichment for guides that score into this precise location, which you can see a magnification down here. And this effect on proliferation exerted by these sgRNAs tracks with a decreased expression of estrogen receptor. And satisfyingly enough, this is an enhancer element. This is marked by K27 acetylation, which is a marker for enhancers, and it's chromatin accessible as measured by ATAC-seq. And notably in this locus, this is a prime element that we observed to be functional on top of one of the transcription star sites of estrogen receptor. We've done the same exercise into the cyclin D1 locus, as I mentioned. This is a pretty big locus of 2.5 megabases. And here we notice three main regulatory elements that were critical for proliferation in the red track and in modulating cyclin D1 expression as measured by the GFP reporter. In particular, we found two enhancer elements, one at 500 kb upstream of the gene, and another one at 125 kb upstream. And we also found a repressive element that when we de-repress it with our CRISPR-I system, we could see an increase in proliferation and an increase in the expression of cyclin D1. So this tells us that essentially out of this big loci, there were very few elements that are necessary for driving expression of this couple of genes. And we can find it by functional genomics. Importantly, we look at the activity status of these regulatory elements in estrogen receptor positive cell lines and patient-derived xenografts, as well as triple negative breast cancer cell lines and PTXs. And we observed that when we bind the sgRNAs according to their effect on proliferation, so here on the right-hand side of each panel, you will see the sgRNAs that give an effect on proliferation, likely by targeting ER or cyclin D1. These elements have an increased labeling by K27 acetylation, specifically in estrogen receptor positive cell lines and patient-derived xenografts. Again, indicating that these are very specific regulatory elements for this disease and they contribute to the expression of these key genes. So this is summarizing the first part. We notice that there's really a minimal set of regulatory elements, essentially one being a major driver of estrogen receptor expression. And consequently, I haven't shown this data, there's a few elements in the cyclin D1 locus which are bound by estrogen receptor. They are in 3D spatial proximity to cyclin D1, and two of them are positive contributor to the expression of cyclin D1, while one is a repressive element, which is trying to counteract and fine-tune the expression of cyclin D1 in this locus. So we thought this was quite interesting in terms of analyzing a single locus, but we wanted to go really genome-wide. And the reason for this is because we are so used at performing ChIP-seq in order to map where transcription factors bind in the genome. And you can see in this genome browser snapshot again that you can find several peaks, for example, for estrogen receptor and its cofactors FOXA1, GATA3. And you see that these peaks have different intensity. There are around 15,000 estrogen receptor binding sites in the genome, but what we don't know is which ones of these are critical. We don't know if, for example, signal of a ChIP-seq necessarily tracks with functionality. It's difficult, as Anthony mentioned, to assign which genes they control because we are always looking at the genome in linear space. And what we don't understand how they are regulating such genes. So how does this element regulate, for example, cyclin D1 downstream, and what's the interplay with other transcription factors that co-occupy a certain locus? So we decided to go genome-wide and build now a custom library, again with a CRISPR-I system, in order to interrogate every single estrogen receptor peak or binding site in the genome. And so this library was targeting all the 15,000 peaks in the genome. It was around 80,000 sgRNAs. And in this case, we've tracked proliferation scoring, knowing that if we would target a critical regulatory elements, MCS7 breast cancer cells would die upon the sgRNA treatment. So as I said, there are around 15K binding sites of estrogen receptor in the genome. And to our surprise, there were very few that were necessary for the proliferation driven by estrogen receptor. These were around 300. And this prompted us to think that maybe ChIP-seq is just telling us all the possible binding sites, and estrogen receptor is kind of engaging different elements for different phenotypes, or some of them have really, they display some functional redundancy, which we're not observing with our screen. And so we were really focusing on these 300 top sites in order to understand what were their features, because we did know that these were the strongest elements in driving proliferation. And so the way we approached this was first to compare our screening data with ChIP-seq signal strength by performing profiling by ChIP-seq on many histone modifications as well as transcription factors. So we divided the ER binding sites between distal sites, so 5 kb away from transcription start site, and proximal sites, so promoters. We binned the sgRNAs, or actually the sites, based on their impact on proliferation. So the bottom row is the top scoring in our screen. And then we look at the signal by ChIP-seq for all these factors. Importantly, when you look at distal sites, the most critical ER proliferative elements were not significantly enriched with histone modifications that occur at an answer element. So we didn't see a statistical significant enrichment in K27 acetylation and K4 monomethylation. So this tells us that just looking at profiling in answer marks, this cannot inform the functionality of the element. Importantly, estrogen receptor itself does not really track with sensitivity. So if we were to slice our ChIP-seq data, taking the top ER peaks, we would have not found the ones that were important for proliferation. But rather what we observed was an enrichment for two transcription factors, which are known to associate with estrogen receptor, such as GATA3 and TFAP2C, to be enriched at distal elements which are bound by estrogen receptors and are important for ER-driven proliferation. And so we believe that now the type of cofactors that are engaged by given transcription factors is critical for determining the functions of those regulatory elements. So not only we saw an enrichment for two transcription factors, we also look at genetics. So we interrogated the pan-cancer analysis of whole genome dataset. This is a collection of 2,300 whole genome sequencing data. And we observed that at the regulatory elements that were found to be most important for proliferation, elements bound by estrogen receptor, we saw statistically significant enrichment in single nucleotide variants in breast cancer as well as other tumors. indicating that not only these elements are functionally relevant, but there's a selective pressure in tumors for modifying genetically these distal elements. And we are investigating what is the functional consequence of these mutations. However, we found the elements in the genome, and now we don't know which genes, how do they engage genes, and how do they, what are the genes that they regulate. And so to do this, we decided to combine our functional genomic screens with single-cell RNA sequencing in a technique called crop-seq, where essentially we can do pooled CRISPR screen, similarly to what we have done before, but read out RNA-seq for each perturbation that we introduce in cells, and coupling this to Hi-C maps, which Anthony just explained, in order to obtain this sort of multi-omic data sets where we have our 3D genome maps with the so-called pyramids that Anthony showed. We perturb specific sites in the genome, for example, site number 1 and number 2 here, or the transcription start site. We can see how these sites are looped in the genome by deconvoluting our 3D maps, calling significant 3D contacts, and reading out how each perturbation, here, for example, TSS, site 1 or site 2, leads to regulation of the genes that are within 4 megabase distance from a given regulatory element. And so I'm showing here an example in the cyclin D1 locus where we do see that site 1 and site 2 engage in the space the transcription start site of cyclin D1, and consequently, when we perturb site 1, 2, or the TSS, the main gene that is regulated is cyclin D1 in single-cell RNA-seq, and we know that this is a dependency from functional genomic screens. And so we took all our hits and decided to screen them in this with this strategy, and to understand what are the genes that are regulated by our regulatory elements. And so one of the example that was particularly striking was the locus of GATA3. I remind you, this is one of the transcription factor which is enriched at the functional regulatory element that we identified. And we noticed there was one element at 1.1 megabase downstream of GATA3, which is in 3D proximity with the GATA3 transcription start site here on the left-hand side. We see it both by Hi-C as well as H3K27 acetyl high chip, because this site is an enhancer element, is bound by ER, FOXA1, and GATA3, and is chromatin accessible. And indeed, by single-cell RNA sequencing, we did notice that by perturbing this very distal regulatory element, the main gene that gets regulated is GATA3. And this tells us that basically we can perturb an enhancer and obtain a phenotype that recapitulates what we observe when we knock down GATA3 in functional genomic screens, and we can assign now regulatory element to the proper target gene. The second gene that we were particularly interested in is TFAP2C. This is the other transcription factor that we found to be enriched at functional ER binding sites. The element, the regulatory element that's scored in our screen was much closer this time, is around 25 kbs upstream of the TFAP2C TSS. And we had actually enough resolution to be able to see loops, even with this short distance. In this case, again, both with Hi-C and high chip for K27 acetyl. And again, this is an element that is bound by ER, FOXA1, and GATA3, and is K27 acetyl positive. And this is indeed an enhancer, because when we, as I showed before, when we knock down either this regulatory element, which we now called TET for transcription enhancer of TFAP2C, we decrease TFAP2C to the same extent as targeting the transcription start site. This is indeed an enhancer element because we can clone it in reporter assays. We do see that it has a estrogen-dependent activation, again indicating it's a ER binding site, and it works in any orientation. So going through all these data sets and taking all our hits sort of helped us to build a map illustrating how ER essentially is branching out to other transcription factors in order to exert its oncogenic program. So we have seen several elements that not only regulate proliferative genes such as cyclin D1, but also regulatory elements that control transcription factors such as GATA3, TFAP2C. We also find them in the MYC locus, which Anthony described before. And this is a way for ER really to promote basically all the necessary functions to drive proliferation in these breast cancer cells. So out of the different targets of ER that we found, we were particularly intrigued by TFAP2C for a few reasons. On one side, when we knock down TFAP2C or its enhancer, and we compare by RNA-seq the genes regulated by TFAP2C or estrogen receptor, we do see a significant overlap between the two, indicating that really these two proteins are in the same pathway. Importantly, if you take luminal A, ER-positive breast cancer patients, and you segregate them based on TFAP2C low or high expression, we do see that the patients that have higher expression of TFAP2C have poorer prognosis, further supporting the idea that TFAP2C is really a critical gene, a critical target that we could consider for therapeutics. So just summarizing this data, we found TET as our prime target into the study. This is an enhancer of TFAP2C, which is bound by ER. It drives expression of TFAP2C. And now TFAP2C, together with estrogen receptor, engages critical regulatory elements which control target genes of estrogen receptor. Importantly, additional facts about TFAP2C. This is a cofactor of ER from public data. We know it's a dependency from functional genomic screens. I haven't showed here, but TFAP2C mainly regulates metabolic genes besides the estrogen receptor genes. And indeed, there's this subset of critical ER targets which are regulated by TFAP2C. Importantly, it stratifies estrogen receptor positive breast cancer survival. And we observed that the TFAP2C motif is enriched at estrogen receptor binding sites, which are engaged upon resistance to modulators or degraders of estrogen receptor. So we're starting to think that TFAP2C could be important also as a resistance mechanism to this kind of agents in the clinic. So I want to conclude here by thanking the people in the lab, particularly Rui Lopes, an extremely talented postdoc who spearheaded this entire work, working very closely with computational biologists Kathleen Sprowski and Saibin Sheng for HiC and CropSeq data analysis, and several people at the Novartis Institute for Biomedical Research, going from drug discovery, data science, particularly our colleagues in the genomics team for wonderful support. And of course, also some collaborators, whether they are internal or external. And I want to thank you again for listening to me and Arima again for the kind invitation. And I'll be happy to take questions.
Perfect. Thanks, Giorgio. Really appreciate the talk. I wrote down a number of different types of techniques that you guys are using, CRISPR screens, of course, going into ChIP-seq, CropSeq, HiC. Life seems pretty interesting there in Novartis and Basel. We have a few questions here that I'll pull up. We have another 10 minutes or so for Q&A, so just another reminder to any of your questions at the bottom of your screen in that Q&A box, and we'll get them onto our speakers. Maybe a couple just to start with you, Giorgio. So going back towards the beginning of your presentation, we have a question around how large, from a base pair perspective, were these 15,000 ER peaks?
So this is a little tricky, right? Because, okay, by ChIP-seq, an ER peak is, depending on the quality of the ChIP-seq, we're around 500 base pairs to maximum 1 kb. If you do cut and tag, obviously it gets slimmer. Maybe one thing that is important to say is that the CRISPR-I window of repression is similarly around 500 base pairs to 1 kb.
Okay, perfect. Oh, we have a few more questions coming, which is great to see. Next one, also for yourself. Given that we have found a specific non-coding regulatory elements that are involved in the tumorigenesis process, is there any therapeutic approach available to target this element specifically in tumor cells? For example, tumor-specific CRISPR-I.
Yeah, I mean, as you know, CRISPR therapeutics is a bit at the beginning at the moment. There are some good cases that companies are trying to implement, and some of them are working, maybe in some monogenic diseases, for example. So I think, these studies are setting the stage for thinking about how can we apply this kind of therapies. In cancer is a little bit more tricky than some monogenic disease, but let's say at the moment we're not there yet, but you need to start somewhere.
More work to do. And this question might be one for both of you, gentlemen. Wonderful talk, so some feedback there, which is great. This person's curious about what factors should be considered while integrating single-cell RNA-seq, ChIP-seq, and Hi-C data. So maybe Georgia, your take, and then I'd be curious if Anthony has anything to contribute there as well.
So integrating Hi-C with ChIP-seq single-cell RNA sequencing.
Just a few reads to put together there.
Yeah. So let's say, first of all, I have to say that this analysis were quite challenging, and kudos to our colleagues who managed to do it. Single-cell RNA sequencing is intrinsically more noisy than bulk RNA sequencing. You just have less steps. So that's why we ended up doing guided analysis looking at genes within a 4 megabase. So we use Hi-C to be guided towards which genes should we look at in our single-cell RNA-seq, knowing that some of them we would not detect them for the technique. So that's, I think, one aspect to consider. The other point, which was particularly challenging for us, we are perturbing enhancers. So the regulation of gene expression is not as prominent as, let's say, knocking out a gene using their coding sequence. So let's say doing unbiased analysis was very, very tough, but using the Hi-C to guide us more or less on what were, getting guided on the TADs, the topologically associated domain, helped us to narrow down the window of the single-cell RNA-seq data.
I myself on mute there, haven't done that in a few months. I was going to say, Anthony, anything to add there from looking at multiple data sets?
No, I agree with what Giorgio said, and just there are many cases where it would be nice if you could resolve the Hi-C contact maps in a pseudo bulk fashion, like how single-cell RNA sequencing is often analyzed. Certainly that would add a little bit more clarity, but seems like just sort of at the very cusp of being able to do that from a technology point of view, and exciting where that might go in the future.
And quality matters a lot. So we need to be very careful on, resolution of your Hi-C data. We were lucky enough that we could use, so we use quite a lot of reads, but less than a usual Hi-C. So we could get less than 5 kb resolution with 600 million reads or a bit more, which was quite good, as well as single cell sequencing. We push a little bit the sequencing to get as deep as we could.
Okay, great. Let's see, a couple more questions here. Keep them rolling. We have 5 more minutes. Do you think multiple binding sites are required in a collaborative manner for the cell fitness? That's why only a small number are identified when you guide individually.
Short answer, yes, absolutely. And this, if you want, our screen doesn't account for that. What we are thinking maybe is to take a certain loci that we care about and maybe screen them in a combinatorial, but obviously there's some limitations to numbers, right? At some point it can get massive, but spot on question indeed. But you could argue on the other side of, if you're thinking about therapeutics, if you're thinking about targeting them, if you're thinking, we were interested in looking at the ones that were the strongest by themselves to drive this phenotype. But yes, absolutely, we are blind to those at the moment.
Some follow-up work. Maybe, another question for both of you. Just as a curiosity, they say, could you share what analysis tools you use for the HiC?
Oh, wow. Anthony. actually, we got recommended from Arima, but maybe here I can be nice. Arima followed us a little bit on the analysis because it was the first time we did it. So we got quite some recommendations from Arima on how to implement a HiC pipeline.
Yeah,
glad we were able to help. Yeah, Anthony, you want to give a little brief overview?
Yeah, I think over the years, we've been supporting many different tools in terms of data pre-processing, feature annotation, like identifying chromatin loops, topological domains, compartments, differential analysis. But I think if you ask us today for initial upfront data processing, we're pointing most people to the juicer pipeline developed by the Aiden lab at Baylor. And that integrates really nicely with JuiceBox, which is a desktop or a web-based software for data visualization. And you can nicely overlay annotations for loops and tags that you've identified using those tools. It also enables you to visualize one-dimensional tracks like ChIP-seq and RNA-seq paired with the HiC maps and the feature annotations from the HiC data. You can quickly toggle across resolutions, like Giorgio was saying, 5 kb bin. You could switch to 10 kb bin sort of in a blink of an eye, and it changes the HiC heat map to a different resolution. For differential analysis, I would say it's there are tools out there, and it's very, very tool specific. We've used a program in the past that we used in a collaborative publication with Kelly Fraser's lab at UCSD is this tool called Diff-HiC, which has lots of useful features for differential analysis, including intra-sample normalization, using matrix balancing approaches, as well as accounting for biological and technical variability between replicates. And it also has normalization strategies for cross-sample analysis. And it's really nice because you get kind of like a differential, quote unquote, loop analysis, as well as more subtle differential interactions that are really founded on a statistical basis, rather than just sort of, is it present, yes or no? So Diff-HiC is the one that we've used in the past and is what is on the 4D Nucleome website when it comes to differential HiC analysis.
Yeah, we use as well Juicer. For visualization, we use HiGlass just because it was more compatible with our IT infrastructure. Yeah. And then we use Mustache tool for calling significant loops. But there are several.
Yeah.
Definitely. 2 more questions we'll try and squeeze in here. I think a question maybe for you, Anthony. How do you normalize HiC between control and treatment samples?
Yeah, that's what I was just trying to say. Is there's this tool called Diff-HiC that does matrix balancing in samples, biological variability, and then cross-sample normalization using some non-linear Loess regression-based approaches. But it's really tool dependent. There's other tools out there like HiC Compare and others, multi-HiC Compare. So I think it just sort of depends on which flavor. But we've used Diff-HiC in the past.
Okay, great. Yeah, the right tool for the right question. And if we can help anyone out there, like we helped Giorgio's lab, please let us know via our website. We have a little email link. You can reach out to our support team. Last question. I know it's 1 minute over. Thanks for sticking with us. Did you do cross-linked ChIP-seq or native ChIP-seq? If cross-linked, did you investigate CTCF as the target? And what was the result compared to the HiC data?
Yes, so the answer is, so mostly cross-linked. ChIP-seqs, we have done CTCF as well. Actually, the reviewer asked to look at CTCF. I have to say it was remarkable to look at how well, so we overlaid HiC data, significant contacts, CTCF ChIP-seq signal, and CTCF motif according if they were convergent or divergent in direction. And it was actually quite remarkable on how well significant contacts were enriched with CTCF binding at convergent motifs. So it was actually quite fun to look at it. So I think it works very well, at least with cross-linked ChIP-seqs. We haven't done much native ChIP-seq in this case.
Okay, great. I think that's all the questions that I can see on the screen. Just wanted to thank you, Giorgio. Great to see you. Appreciate your time, and really great to have the opportunity to work with your group further. Thanks for sharing your story. Anthony, always good to have a peek behind the curtains at Arima. Thanks for the overview, and thanks for addressing the question, gents. We appreciate your time. Thank you all. Have a nice day. Cheers.